Hi everyone. My name is Mitul. I am the chair of the Research and Innovation Committee of the Institute of Psychiatry, Psychology and Neuroscience, and we are pleased to be hosting this event on neurotechnologies, current developments, applications and ethical issues. The Research and Innovation Committee is a committee led by staff, for staff, and we focus on issues such as career development, quality of the research environment and culture, and also innovative strategies to improve our potential research and impact. And this has led to the development of an innovation subgroup, which is led by Marija Petrinovic, who I will introduce in a moment, who is my co-host, my co-chair. So we are recording this meeting, and so if anyone needs captions, please press the close captions button. So I'll now hand over to Marija, who will introduce the actual event. Many thanks for joining.
Thank you, Mitul, and thank you everybody for joining this really exciting two-day event, during which Mitul and I will be your hosts and chairs. And during this two-day virtual, unfortunately, or maybe for some fortunately, virtual conference, we will be bringing together some leading experts at the forefront of neurotechnology developments and discuss some cutting-edge innovations which are changing the face of neurobiological research today. During these 2 afternoons, we will focus on neurotechnologies for neuroscience research, mental health, neurology applications, and we will cover advances from molecular, optical, electrophysiological, and up to neuroimaging approaches. We will also discuss ethics and applications of these technologies for treatments of a very wide range of mental health and neurological conditions, including, for example, movement-related disorders, spinal cord injury, epilepsy, Parkinson's disease, depression, and so on. We are encouraging questions from the audience, and all the questions we will take after each talk. So if you have a question, please write it in the chat and we will ask those questions on your behalf after each talk. Here you can see timetable for day 1. It's a very packed event afternoon with very exciting talks about iPSCs and so on and so on. So please stay with us. On day 2, we will also have really great talks with 2 keynotes coming from Ed Boyden and Dustin Tyler, and a series of short talks. So please also join us tomorrow for the talks and for the panel discussion after the talks at the end of the event. And without any further ado, I would actually now like to welcome our first speaker who will deliver the opening talk. This is Charlie Winkford-Smith from the Innovate UK Knowledge Transfer Network. Thank you, Charlie, for joining us today.
Sorry, can you see the presentation?
Yes, Charlie, thank you.
Brilliant. Sorry for the delay. Teams seems to kill my computer for some reason. But yeah, thank you very much for the opportunity to present today. I'm from the Knowledge Transfer Network, KTN, which for any of you who are unfamiliar with us, we're predominantly funded by Innovate UK and the research councils. And our role is to build communities and create connections between innovators. So we've got teams in agri-food, digital, transport, health. We really cover all sectors. I'm in the emerging technologies team, and our main focus is on new technologies coming out of universities. I lead the Neurotechnology Innovation Network, and I also lead on our work on in silico medicine. So I've been asked to give an overview of the neurotechnology landscape in the UK, but I thought it might be worthwhile. Ooh, there we are. Sorry, slides going slowly. I thought it might be worthwhile to give a quick overview of what's going on globally as well. So around the world, there are some large-scale government strategic investments in brain-related R&D. Probably the biggest in the world is the Brain Initiative in the US, where they've invested over 1.7 billion since 2013, with an estimated $400 million a year to be invested until 2023. In the EU, there's the Human Brain Project, with an investment of around €1 billion over 10 years that was launched in 2013. And around the world, there are other investments, for example, in Canada, Australia, Korea, and Japan. In China, there is definitely a lot of investment, but it's much harder to put a number on the size of that investment. I think it's interesting if we compare investment scale versus GDP, and with that you get a slightly different picture. So Australia, for example, is investing very heavily in neurotechnology. In part, that's because they've seen previous success through the cochlear implant. Korea is also investing heavily in neurotechnology, as they see it is an important way to combat a rapidly aging population. As you can see, the UK is a bit further down this list, in part because we haven't thus far had any specific funding for neurotechnology. However, UKRI has been investing in neurotech. So over the last 10 years, there's been approximately £9 million a year invested, predominantly by the research councils, but Innovate UK has in recent years been investing as well. Excitingly though, EPSRC and MRC recently announced a £6 million call for neurotechnology networks. This call has just closed, but we should expect the results in March. From the bids I've seen, there are some really exciting network applications that have gone in, so it'd be brilliant when these networks launch next year. If we look at the regional distribution of companies around the world, again, the US is leading the way. This is in part because of the large government investment through the Brain Initiative, but also through DARPA, their Defense Research Funding Agency. So some of the most well-known neurotech companies are based in the US. Neuralink, for example, is Elon Musk's company. Even companies that were actually started elsewhere, so Dream, for example, has now moved to the US and is based in Silicon Valley. Synchron, which was based on research from the University of Melbourne, is also based in the US. And it's also worth pointing out that big tech is very interested in neurotechnology. So last year or two years ago, Facebook bought Control Labs, but Amazon, Microsoft and Google are all investing heavily in neurotechnology. The figure on the screen at the moment is from a US report from last year. It lists the top 200 neurotech companies in the world. I perhaps take it with a pinch of salt. There are certainly a lot more than 13 neurotech companies in the UK. But it does show that there are neurotechnology companies popping up all over the world. Again, I'd stress that there is a lot going on in China, but it's perhaps harder to pinpoint those companies. Last year, we put together an interactive landscape map for neurotechnology organisations in the UK. So we identified over 200 organisations, and you can search through this landscape map based on capability or location. We've also put together all of the UKRI funded projects. So this is around £100 million worth of grant funding over 10 years and 251 projects. Again, you can search this by either funder, organisation or type of technology. So do go and have a look at that when you have a chance. So if we just look at the UK landscape in a bit more detail, of course, the UK has some really world-leading universities looking into neurotechnology, covering areas like brain-computer interfaces, deep brain stimulators, bioelectronic medicines, and lots of other types of neurotechnology. There are a small but growing number of neurotechnology companies now in the UK. Probably the most well-known of this group is Galvani Bioelectronics, which is a joint venture between GSK and Verily Life Sciences, which is an alphabet company. So they've invested around £540 million over 7 years, so a very significant investment. And I'll go into a bit more detail about some of these companies in a minute. There is also a growing ecosystem of support for neurotechnology companies and academics in the UK. So whether you're looking for support in new materials or general commercialization advice, and again, I'll go into a bit more detail about those in a minute. So I thought it'd be useful to just give some examples of UK neurotechnology companies. And I've also put on the screen here some of the key diseases that these companies are trying to target. So Bioinduction, based in Bristol, have developed a deep brain stimulator device called PicoStim, which is much smaller than current types of devices. The battery is located just underneath the skull, and that means that you don't have to pass wires all the way down through the neck. This actually results in halving surgery times, so it's a much more cost-effective way to treat Parkinson's disease. But they're also looking to build this up as a platform device for other diseases as well. Cumulus Neuroscience, which was previously known as Brainwave Bank, is based in Northern Ireland. They've developed a wearable EEG headset that, when used in combination with brain training type games on an iPad, you can measure cognitive ability and potentially identify Alzheimer's at a much earlier stage than conventional measurements. MagStim are probably the world leader in transcranial magnetic stimulation, which is used to treat depression, and in particular drug-resistant depression. Their current treatment takes about 37 minutes, but they're developing a new device which can deliver that same treatment in 3 minutes in an outpatient setting. Neurotechnologies is a spin-out from Ulster University. They've developed a device called Flex EEG, which is a wearable headset, EEG headset, which is completely wireless and just fits within a baseball cap. There are all sorts of applications for this. They've also developed a software suite called NeuroPrecise, which helps analyze the information from this device. One of the key areas that they're trying to target initially is stroke rehabilitation. Galvani Bioelectronics, who I mentioned previously, they're developing bioelectronic medicines, which are tiny electrical devices that fit around a nerve. Galvani are targeting the vagus nerve and looking into rheumatoid arthritis initially. BIOS Health are based in Cambridge. They also are targeting the peripheral nervous system and are particularly interested in circulatory disease. We've put together a couple of videos which showcase some of these companies. So we've got videos on Neurotechnologies, BIOS and MagStim. Do go and have a look at these. They're on YouTube, and just give a bit more insight into what these companies are working on at the moment. But of course, neurotech is not just about health. There are lots of non-medical opportunities for neurotechnology. Gaming, for example, is a really exciting area for neurotechnology, particularly brain-computer interfaces when combined with VR. There are other opportunities maybe in sports. In education, for example, there's a spin out from Oxford University called Chronotype, where they're developing a wearable brain stimulator headset, which they say can enhance cognition. And of course, there are a lot of applications in defense and security as well. So I would just like to draw your attention to this report, which was published by the MOD in May this year. The MOD and the government in general take a broader view of this area. So they look at human augmentation. So this is neurotechnology, but also things like genetic engineering, exoskeletons, augmented reality, and other areas. And again, they're not just interested in getting back to baseline or biological potential. They're really interested in human enhancement as well, and the implications that that might have. I would emphasize, if any of you are developing neurotechnology devices, to consider cybersecurity at an early stage. It's much easier to embed cybersecurity at an early stage rather than as an afterthought. And it's something of interest and concern to security services. I would also emphasize, if you're developing a neurotechnology device, do think about the defense and security applications of that device, because there may well be funding in the future for that type of device. As I mentioned, there's also a growing supportive ecosystem for both companies and academics doing neurotechnology. The Henry Royce Institute up in Manchester is the National Institute for Advanced Materials Research. They've got a real interest in new materials for bioelectronics. The manufacture of active implanted surgical instruments facility, the Mehta facility, is a collaboration between King's, UCL and Newcastle, and is a new clean room manufacturing facility that's located at St Thomas's Hospital. They have the ability to manufacture Class 2 and Class 3 active implanted devices. So it's a fantastic resource for academics and companies. The Centre for Process Innovation, CPI, has a huge amount of experience in helping companies commercialize new wearable medical devices. And likewise, Mindtech up in Nottingham has a huge amount of experience in helping companies commercialize new devices for mental health care applications. We've also published a couple of reports recently. So last year we published one called Towards a UK Neurotechnology Strategy. This gives much more of an overview of the UK neurotechnology landscape and a bit more information about some of the potential applications of neurotechnology. This year, in June, we published a roadmap for neurotechnology in the UK, where we set out key recommendations that we believe, if implemented, would be absolutely transformative for the sector in the UK. But as I mentioned right at the start, KTN is here to connect innovators with new partners and opportunities. So if you are developing a new neurotechnology device and you're looking for partners, whether that's in academia or in industry, please do get in touch with me. It'd be fantastic to find out about what you're working on, and hopefully we can make some useful connections for you. So I will leave it there, but thank you very much.
Thank you very much, Charlie. This was a really interesting talk, and what I was constantly hearing you repeating is that, you know, there is so much interest in enhancement of human cognition and in general kind of a human enhancement. So it would be really interesting to hear the discussion on the ethics of all of these applications, and this will be discussed then tomorrow during our panel session. Thank you very much again, and now I would like to announce our first keynote speaker, who is and this is a Professor Michael Nietzsche from Leibniz Research Center for Working Environment and Human Factors. He will be telling us about optimization of transcranial electrical stimulation. Michael, are you with us?
Okay.
Martin. Oh, you're with us. Great. Thank you, Michael.
So now I'm in. Okay, so yeah, many thanks for the invitation. It's a great pleasure. Unfortunately, it's all virtual now, and hope can come back soon to London. Okay. Thank you very much. So I want to talk the next 15-20 minutes or so about optimization of transcranial electrical stimulation. So as next slide, please. Okay. So as you all know there are many non-invasive brain stimulation methods which have been introduced in the last 20-30 years. We can now, with non-invasive stimulation, induce activity of the underlying cortical area with transcranial magnetic stimulation. We can also induce neuroplasticity, which means longer lasting alterations of cortical excitability with a couple of techniques. This includes repetitive transcranial magnetic stimulation, paired associative stimulation, and transcranial diachronic stimulation. You can see in the lower panel that when you stimulate the motor cortex, for example, with RTMS, here a special version of RTMS, you can enhance or reduce excitability for prolonged time courses. And you can also modulate cortical oscillatory activity with techniques such as transcranial alternating current stimulation. In the next minutes, I want to concentrate on TDCS and TACS. Next slide, please. And so we have developed the technique, how it's used currently. About 20 years ago, there were early animal experiments and also some single studies of other groups. The principal mechanism of action is that you lead small currents, so intensity between 1 to 3 milliampere currently, over the scalp to the brain. And these currents are too small to induce action potentials, but they modulate resting membrane potentials. And you can imagine that when you, at the subthreshold level, depolarize your resting neuronal membrane potentials, then it's easier to induce action potentials, whereas when you hyperpolarize and you reduce spontaneous activity and excitability of the underlying tissue. This worked relatively well. So you see on the right side here some results of early experiments from our group, where we have shown its stimulation for a few minutes. On the upper figure, stimulation between 5 and 30 minutes with the anode over the primary motor cortex, that you can enhance cortical excitability as measured by motor evoked potentials for up to an hour or a bit longer after the end of stimulation. Whereas with the cathode over the motor cortex, so reverse polarities, you reduce cortical excitability, and also these effects can last for about 1 hour in the classical protocols. Next slide, please. So we got these results, but especially, of course, for clinical application, you want to have longer lasting effects, and the question is how to optimize stimulation effects. So the question is, are the effects in any case linear? So does stronger, longer stimulation get you better effects? Do you get always the same effects everywhere in different cortical areas? And at the end, how to optimize protocols? Next, please. To think about optimization of the effects of the stimulation technique, you have to learn something about the physiological background. And what we have found out by a couple of early studies, pharmacological and non-pharmacological studies, is that we induce neuroplasticity of glutamatergic synapses and in a way that we activate NMDA receptors, which control calcium influx to a target neuron, and depending on the amount of calcium influx, this results at the end in removal or insertion of AMPA receptors to or from the subsynaptic membrane, and this strengthens or weakens, respectively, synaptic connections by controlling the depolarization of the subsynaptic membrane. We have shown that NMDA receptors are critically involved via experiments where we combined transcranial current stimulation with dextromethorphan. You can see this on the right side, on the right upper figure, that when we applied dextromethorphan, then we didn't get any plasticity in the primary motor cortex as a model region via anodal and cathodal TDCS. So NMDA receptor activation is crucial. And we have, as shown in the lower figure, also shown that calcium is involved, because when we block calcium channels with flunarizine, you can see that the after effects, the excitability enhancing after effects of anodal stimulation are abolished. So we have a kind of glutamatergic calcium-dependent plasticity, which resembles quite nicely predominant versions of plasticity as detected and described first in animal experiments. Next slide, please. Then the question is, what does this tell us about dosing of stimulation? Because the effects are calcium dependent, and I've shown you that before, that low calcium concentration will induce removal of AMPA receptors and thus should result in LTD-like plasticity, whereas higher concentration results in LTP-like plasticity. You have a transition zone, so-called no man's land 1, and where you don't see clearly directed plasticity, and calcium overflow is also prevented when calcium concentration is too high. This is no man's land 2, whereas there's also restriction of plasticity. On one hand, this means that as long as you're in a certain range of calcium concentration, you will enhance the efficiency of stimulation to induce neuroplasticity. We have shown this for anodal TDCS. On the right side of the slide, you can see that enhancing stimulation intensity between 2 and 1 milliamp enhances efficiency of TDCS, and also enhancing duration of stimulation with the intensity of 1 milliamp. That's the lower figure. You can see that enhanced duration of stimulation improves also the efficiency of TDCS. Next slide, please. But these calcium dynamics also imply that with specific stimulation protocols, you might not get your expected plasticity effects. And we have, as you can see from the figures on the right side, doubled stimulation duration as compared to our standard protocols from 13 to 26 minute stimulation. This was done by Katja Montesilva some years ago. And you can see here that we don't get the expected excitability enhancement with anodal stimulation anymore, but a reversal of the effects into excitability diminution. We expected that this was due to calcium overflow, so that we were in no man's land 2, and so we explored this mechanism via blocking calcium channels again, via flunarizine, and you see in the lower figure that its reversal, at least, was abolished by calcium reduction. So we have nonlinear effects also when we overdose. And then next slide, please. The question is, of course, how can we optimize stimulation parameters? How can we optimize dosages to improve efficiency of TDCS? And there are a couple of options to determine directionality, regional restriction, and duration of the effects. Can do this via maybe further prolongation of stimulation, via stimulating the stronger intensities, maybe repetitive stimulation, of also pharmacological interventions. We have worked in all of these fields, but I want to show you just some single important results of experiments. Next slide, please. And this is a study which has been published 2 years ago, where Desmond Akbuada titrated stimulation dosage for intensity between 1 and 3 milliamp and for stimulation duration between 15 minutes and 20 minutes. So 15 minutes being the standard protocol, 20 minutes medium protocol, and the maximum duration was 30 minutes of stimulation. On the right side of the slide, you can see the results we get. So with all intensity and duration combinations, we got excitability enhancement with anodal stimulation. You can see this on the upper part for 1 milliamps and 2 milliamps and 3 milliamp. But when you look at the amplitude of the effects, then you can see that 3 milliamp was gradually better with respect to the magnitude of the after effects. Next slide, please. It is a systematic titration study also for cathodal TDCS, and what we have seen here is that upper part is again 1 milliamp, lower part 3 milliamp, in the middle the 2 milliamp condition. As the effect was a bit more of a prolonging stimulation duration, enhancing stimulation intensity was a bit different. So we saw the effects again with 1 milliamp, with fairly standard protocols. With 2 milliamp, we didn't see an excitability diminution, but an excitability enhancement. Now, this might be the case because with 2 milliamp you stimulate stronger, you might increase calcium over the range, which induces LTD-like plasticity. So you get into the LTP induction range here. You see this quite prominent for 2 milliamp 20-minute stimulation. And with 3 milliamp stimulation, however, you get again an excitability diminution. We looked a bit into the mechanisms, thought that with 3 milliamp you then might be even over the range of the induction of long-term potentiation, like so calcium overflow, counter-regulatory mechanisms could apply. So we tested this via different dosages of calcium channel block and could indeed see with certain dosages then a reversal of this secondary inhibition into an excitability enhancement. So this shows that it has to be taken into account when you optimize the protocols, that enhancement of stimulation dosage not only results in linear effects. Next slide, please. So I've shown you so far that we get dosage-dependent effects of TDCS, which are, however, not linear in each case. And however, extending TDCS beyond 1 milliamp in 50-minute stimulation can gradually improve the efficiency. Next slide, please. So all results I've shown you so far were for the primary motor cortex, and then of course it's a crucial question if you think about application, do we see these effects or similar effects also in other cortical areas? I want to show you some data about newly conducted studies where we did dosatory prefrontal stimulation and monitored cortical excitability autoreactions induced by TDCS with cortical evoked potentials in the EEG. And here we compared prefrontal with motor cortex stimulation effects. Next slide, please. I want to show you the data we obtained for anodose stimulation. It's known that for these cortical evoked potentials, that different parts of these potentials are dependent on different pharmacological mechanisms. So here on the left side, the effects we got for the primary motor cortex. On the right side, we got for the prefrontal cortex. We stimulate, we did TDCS with low and with high dosages. And you can see here that in the primary and in the prefrontal cortex, there's a major effect on early potentials, which depend on the glutamatergic system. You see this for the P30 wave, and you see similar effects also for the P60 wave, which depends on glutamate and on GABA. You see also in both cortical areas a reduction of a wave, of the amplitude of a wave, which depends on GABA, which fits nicely to GABA reducing effects of TDCS we have seen. So the effects are relatively comparable. Next slide, please. And this shows that the M1 effects are also principally transferable to prefrontal areas. However, the effects might be gradually smaller. Next slide, please. Another important question also relevant for optimization of the efficiency of stimulation is if the stimulation has similar or the same effects in all individuals. And it's known, and it's also self-evident for neuromodulation, that we have effects which depend on the state and on enduring properties of the cerebral cortex. We explored this for some aspects. We explored this for higher age in adults. So in this study, we compared plasticity induced by anodal and cathodal TDCS in young subjects, in subjects between 55 and 65 years, and subjects which were over 65 years old. You see for anodal stimulation, right side upper panel, that in young subjects we got the plasticity we saw in earlier studies. There's a gradual decline of the induced plasticity with higher age between 55 and 65, and in subjects over 65 years old with 15-minute stimulation and 1 milliamp in this study, we didn't see any effects or no LTP-like effects in subjects over 65 years old. This was selective for anodal stimulation. For cathodal stimulation, we didn't see these effects. And on the C, we have here sham stimulation, where we have stable MEP amplitudes. So there's a this stimulation protocol, we got reduced plasticity in associated with higher age. Next slide, please. The question is then, of course, can you induce long-term potentiation like plasticity in higher age at all? So does this maybe depend on the specific stimulation protocol, which was relatively weak? And so we did the titration studies we did before in young healthy subjects, we did in older subjects. On the left panel, you have the early elderly, so the subjects between 55 and 65 years. On the right, and you have the subjects over 65 years. You can see here with stimulation between 1 milliamp and 3 milliamp, 15 to 30 minutes. You can indeed, with intensified protocols, you can indeed plasticity also in these age groups. And again, here you can see there's a gradually improved efficiency of the stimulation with amplitude and also with regard to duration with higher dosage of stimulations. Next slide, please. Another aspect which might affect the inducibility of plasticity via non-invasive brain stimulation, including TDCS, is the chronotype. So you all know that there are people who are very awake in the morning and get very tired early in the evening, and there's the other way around. So you have larks and olds. And we asked in this study conducted by Ali Salehinejad if this being morning or evening type affects your plasticity and if this has also functional consequences. Next slide, please. So we applied annual and cathodic TDCS in early and late chronotypes. You can see this on the left side, and as expected in the early chronotypes, they had much better plasticity in the morning as compared to the evening. And this applies for LTP and LTD-like plasticity, whereas in the evening chronotype, these subjects had better plasticity when we induced plasticity in the evening as compared to the morning. So indeed, chronotype affects the efficiency of TDCS. And this has effects also on motor learning. We did a sequence motor learning task where reduced reaction times show better performance. And likewise, we saw that in the early chronotypes, they had better performance in the morning than the evening, whereas this effect was reversed in the late chronotypes. Next slide, please. So individual characteristics affect plasticity response to TDCS. Higher age reduces LTP-like plasticity, but not LTD-like plasticity. However, plasticity can still be induced also in higher age. Chronotype determines the size of plasticity response and the amount of learning at specific daytimes. Next slide, please. So I want very shortly to show you, need just a minute or something like this now, to show you some new TACS protocols. With TACS, you can modulate oscillatory cortical activity. And this has been shown some years ago already, that you can enhance alpha activity for up to 1 hour after the end of stimulation with oscillatory stimulation, so alternating current stimulation at a similar intensity as TDCS. You can also you can also modulate this activity in the visual cortex. The important thing is here you don't induce this oscillatory activity, but you entrain it, so you enhance already present activity. It was shown for the visual cortex, for example, where it was shown when beta frequency is dominant, then the most efficient stimulation to enhance excitability was in the beta frequency range, and when alpha frequency was dominant, so eyes closed, room dark, then you can see that alpha frequency stimulation had better effects. So next slide, please. One drawback of the currently available techniques to modulate oscillatory activity is that with TACS you cannot induce activity, so if not much is there, you won't be able to induce this activity. With rTMS you can induce respective activity, but this is relatively short lasting, so lasts for some minutes. So our aim was to induce long-lasting alterations of oscillatory activity by a combination of rTMS with TACS. And the idea was with rTMS we induce this activity, with TACS we stabilize this. We applied in the principal experiment the TMS pulse at the peak of the TACS sinusoidal wave, and it also have, of course, some control conditions. Next slide, please. And what we have indeed shown that this combination for the alpha frequency, and we saw similar effects also for theta frequency, enhances oscillatory activity in this frequency band. And these effects, this is figure B, lasted for at least 30 minutes after the end of stimulation, were restricted, figure D, to the frequency band we were stimulating. So we've developed here a protocol which can induce activity at a certain frequency band and stabilize it. Next slide, please. So systematic dosing of stimulation parameters reveals partially superior effects of intensified TSCS protocols. Individual characteristics such as age and chronotype affect plasticity response. And I've shown you also that advanced TSCS protocols allow the induction of long-lasting after effects, which are, as we have shown for theta frequency, time is not sufficient to show this here, which are, however, functionally relevant for cognitive processes. Okay, last slide, please. Many thanks for your attention. That's all the people who are doing the work in my laboratory. And time for questions, I guess. Sorry for being a bit too long.
No, you were just on time. Thank you so much for this wonderful talk and very interesting one. We have a few questions for you from the audience.
Sure.
So one question is actually about the LTD. So the question is kind of why higher age does not affect LTD, but affects LTP?
So what is known from animal experiments, so they show exactly the same pattern. And it's not that LTP, also in animal models, is not inducible, but the threshold for the induction of LTP is enhanced. And you can speculate, I mean, you have a reduction of most of neurotransmitters and neuromodulators. And if you think that there's, for example, reduction of glutamate, of glutamatergic synapses, and maybe also of glutamate release, then, and if you think about this calcium dynamics, then if the same presynaptic activation will result in lower glutamate release, then it's with the same level of activation, there's a larger range where you reduce calcium influx, and then there's a larger range of activity which will induce LTD. So that's, I think it's a simplified idea, but in principle, this is how it's thought, how this works at the end. And of course, the numbers of spines is reduced and many other things. But the critical thing, and also in animal experiments, is that it's not, there's no LTP, which will be disaster for old people, of course, but you need some more activity, and you need higher dosing of your intervention protocols to get similar effects as you get in young people.
Mm-hmm. Thank you. And another one of our audience member actually asked, so basically when you do all of those stimulations, you affect the cortex and then subsequently subcortical regions, such as, for example, striatum, right?
There are, at the end, it all depends on dosage. Yeah, if you want to directly stimulate also the striatum, I think you need higher activities. It's also, the architecture is different. So getting to details, now you have to meet long axis of at least the majority of neurons to get relevant effects. This might be more difficult in the striatum, but we have, I talked about plasticity effects today, but we see also prominent connectivity results. So when we stimulate the primary motor cortex, we get an enhancement of the cortical subcortical motor network, and this includes premotor areas, parietal areas, but also striatal areas, and for example, the pallidum. It was in MRI studies by Raphael Polania in our laboratory in Göttingen at this time, quite nicely. So you get network effects of the stimulation, but with our usual intensities, we don't see direct electrical induced, not connectionally, but electrical field induced effects in subcortical regions.
Oh, very interesting. And actually following on from there, our audience members were asking about excitation inhibition imbalance. We all know that excitation inhibition imbalance is thought to be in root of many neurodevelopmental neuropsychiatric disorders, such as, for example, autism spectrum disorder. So do you envisage using of such stimulation in those conditions? And how do you then control for the long-term effects? These are kind of questions summarized into one.
Yeah. So if you think about patient studies, and there are some studies, at least one I have in mind for autism, which has been recently conducted by collaborators, where there's also we discussed the effect on excitation inhibition balance, and so what you see with TDCS in healthy humans is that you enhance with NOTCS glutamatergic plasticity, you reduce GABAergic activity. It's nicely shown by Charlie Steck some years ago in an MRS study. With TDCS, you reduce both glutamatergic activity and you reduce also GABAergic activity. So that's what was shown, at least for the motor cortex. For other regions, some partially different results are seen. So there is, it seems to be there's not an isolated effect on excitation or inhibition, and it has not directly shown at the physiological level what happens if the balance which is there in healthy subjects is no longer intact. And how you counteract this, and I think that's not a trivial question. We have to explore this directly. MRS study might help, and it's the unfortunate thing a bit is that especially in clinical studies, the bulk of the studies, and I think that's not only for TDCS, are mostly restricted to behavior. And then of course you can look also for long-lasting effects, but what we would like to see and what would be important if we talk about optimization is that you get also that you add physiological results, that you add imaging, that you add maybe TMS studies or something like this to see what happens mechanistically, and on this foundation have more targeted stimulation. This is also true for excitation inhibition balance, where you could think about also pharmacologically stratify the effects, for example. Also, this is possible. For example, we have done studies with serotonin uptake inhibitors, where we have shown that in this case, both anal and cathode stimulation induce LTP-like plasticity. And then it goes in a certain direction, and this should more or less selectively also enhance efficiency of the glutamatergic system. And so you might stratify stimulation techniques based on these things, and then also come to advanced protocols. So I think we all won't get bored during the next years.
Thank you so much for this. It's really such an interesting topic, and what you have said, I can't agree more. We need to really combine different technologies kind of at the same time and check what is really happening mechanistically. And it will be also interesting to see how actually our chronobiology actually fits into all of that. Thank you very much, Michael, again. And in the interest of time, unfortunately, we have to move on.
Absolutely. Okay.
Thank you.
And yeah, see you later. Bye-bye.
See you, Michael. Thank you. We now need to move to another talk, very exciting one again. It is Lucia Valmaggio, again from King's College London. Lucia Valmaggio will tell us something about virtual reality in mental health. And Lucia is a reader. For those of you outside UK, this is associate professor in clinical psychology and digital mental health. And she's also head of virtual reality lab at King's. Lucia, thank you so much for joining us today.
Thank you, Maria. Thank you for the introduction and for the invitation to talk to you about the work that we do in our lab. So I don't have nothing to declare. I'm going to talk about what virtual reality is. You already heard a bit about it from Paul, how we use it for mental health, and then show you examples from projects that we have ongoing at the moment to show you how it can be used. One of the reasons why people are using digital technologies, including, for example, virtual reality in mental health care or assessment or research, is that there is a lot of need for mental health treatments, but very few people who can deliver it. So the idea is, especially thinking about the fact that most people who will develop a mental health problem do so at an early age, and they are all digital native. Can we use digital technologies to help them cope or live better with the problems that they have? So can digital technology have a role? I do not have time to go into all the ethical concerns around virtual reality. I just want to make You aware of the fact that there's been a lot of studies going on looking at how ethical it is, but also looking at the safety of using this type of technologies with young people. And it has been found to be suitable for children, teenagers, and adults when delivered in a blended way. So when there is somebody there who knows what they're doing, delivering the intervention. So you already heard a little bit about virtual reality. Just to remind you again, the good thing about virtual reality compared to, for example, non-immersive things like showing something on a 2D screen is that it enhances the sense of presence or the sense of being there. And why is it important is because if your brain thinks that you are in a situation, your physiological response is similar to the one that you have in real life. And we know that if you want, for example, to change the way people respond in a situation, either their feelings or their behavior or their physiological activation, is important that those are activated when you try to do so. At the same time, you know that it's not real. So there is this, this enables you to try things that maybe you will be too scared to do in real life. Often when we see people, we see them either for testing, as you can see here, or for therapy in a situation like this, where the patient comes and tells the therapist what their problems are, for example, or where you assess neurocognitive functioning. The reality, of course, is that the problems happen in these type of situations. So supermarket, work, in the train, a school, in places, you know, that are nowhere near similar to this. So the question is, how do you bring reality into this type of consultation rooms of labs? And that's where digital technology can help. So that's the second term that is important in virtual reality, which is ecological validity. So if you are in a consultation setting or lab, you have high experimental control. If you are in the supermarket, for example, when you're doing exposure in vivo, when you take somebody to a place where they are afraid, you have very, very little control. You don't know who is there. However, the ecological validity, so how similar it is to reality, is very high. And virtual reality enables you to combine both. So it enables me to know how many people are in an environment, how loud the sound is, are they friendly or unfriendly, and at the same time, it feels real. Okay, so that's the beauty of virtual reality, if you like. It enables you to bring reality into the lab. And the way we use it is to assess cognitive functioning in real time, or symptoms as they occur in real time. And in therapy is what is called virtual reality assisted therapy, where you practice with an individual the difficult situations that they face in real life in virtual reality first, so that they can learn new ways to cope with those strategies, and then they can practice, which is of course important still, they can practice the new learn strategies in real life. We have a large lab in the Institute, which looks at different things, from the mechanisms involved in the onset and maintenance of mental health problems, the treatment, prevention, and training. Obviously, in 10 minutes, I can't talk to you about all of them. I've chosen a few examples of things that I think, seeing the topic of today's talk, might be of interest to you. So the first is the use of neuroimaging. Just to let you know that if you are interested in knowing more about how to use it in mechanism, we have published a literature review in, for example, the use of psychosis, and there are quite a few papers coming out in recent years. So majority of papers have come out in the last 5 years. So it's a literature that is growing by the minute. So first, I'll tell you a more typical use of virtual reality before we go into the neuroimaging. And that is where you use the same situation and you modify what people are experiencing. So this is a study we are doing with Marta Di Forti. She's interested in looking at, do different type of cannabis influence people in a different way. So we are showing everybody the same situation. What the variable that is changing is the type of cannabis that they're using. And this is in 2 samples. One is a healthy controls, and the other one is people with a diagnosis of psychosis. And in both groups, people use different types of psychosis. And they wanted, so these are the people we consulted with. We develop everything with people with lived experience. They wanted a grocery shop. So here you can see important, they wanted somebody a bit edgy outside, so they chose police. So here you can see the police people outside. And what you then can see is there is a typical British grocery shop, and then they have to go towards people that are talking to them. Okay, so just to give you a bit of a feeling, and the idea is that they have to do buy certain items or go around and then asking them how they feel and measure the physiological response. The neuropsychology, neuroimaging can also, has always been the next frontier, if you like, because of course you cannot wear the headset in a neuroimaging device or equipment. And this is something that has been developed by Ku Xian, who is in the Center of Developing a Brain and here at King's College. He's published this in Nature, so you can look it up. I just want to show you how it works, because it's been very inventive. So this is for young people, young children, who have to do the neuroimaging. And what is done very smartly is integrated the sound of the scanner in what people see, and also the test that they have to do. So here you can see, obviously it's for children, so the music is quite nice. This is the sound. This is the person controlling with their eyes the images that they see. And as they move into the scanner, they also move physically. And then you will see in a second, I don't have a lot of time, I just want to show you because it's children friendly. You will see that the bear that comes through, and then you have to follow and do all type of games with the bear while you're doing your exercise or your assessment, really, in a neuroimaging. And the brilliant thing about what he has done is this the first time that we have stereoscopic view in a virtual reality environment, in a neuroimaging, without having to wear, you know, a traditional head-mounted display. We publish in Nature, so it's quite novel. And if you are in this field, I will recommend you contact him. So just a couple of minutes left. So very briefly, I'll show you example. It can also be used for therapy. Again, if you're interested in the literature, this has been reviewed. We have a study that just finished with psychosis. We've been doing this work for a longer time. So we are now at the time where we are looking at how you implement it in the clinical practice. This is, for example, work by Matteo Cella, looking at negative symptoms in people with psychosis. And it's just finished and we found good results. But I want to show you again a video of one with eating disorder, because you might have this in your practice. And this is to assess how people respond to eating disorder. And at the same time, together with young people with eating disorder, they asked us to create an environment where there was a fantasy figure that could help them decrease the number of the anxiety level while they were in the virtual reality environment. And you will see it's, oops, if I do it correctly, you will see that.
Think about eating this food.
So this is to
How
assess
anxious do you
it.
feel?
So you can show food or you can show rating scales. Just move it on a bit. And this is the little fantasy elephant that they chose. And here you can see they can interact with food, which is very anxiety provoking for them. With the
use the slider to make a selection,
and also
then press OK to enter the kitchen.
They asked us to have a person that can help
them. You are doing the best you can right now. Nobody is here to judge what you will do in the kitchen.
That you can see here are dialogues that people have who have recovered from eating disorder have said have helped them in the past. So just to say, of course, it's not just positive. There are quite a few key challenges, as you can see here. So synchronizing with physiological measures is still quite difficult. And also it's quite difficult when you want to translate it into daily practice or implement it in clinical care. What you need to do and the skills that people need to have is still quite a challenging thing. Just to we have a fantastic VR developer. Here you can see him, Jerome Di Pietro, is creating all these wonderful environments. And these are the institutes that pay for our work. And thank you for your attention.
Thank you very much, Lucia. Let me just see how we can do this virtual applauses. I can't do it in this version of Teams, but a big applause to you. This is really so great. I'm almost kind of enticed to go into the scanner as one of your volunteers. It seems so interesting. We're a little bit behind the schedule, but there was a very important question that I would really like to ask you. It is about PTSD. I believe that on one of your slides, it was written it could be used for treatment of PTSD as well. Can you use such an approach there? Let me just read the question. Can you use it kind of to erase bad or negative memories and help people with PTSD?
So most of the work that has been done in the field of PTSD has been done by a group that works with war veterans in the States. And they've recreated the typical, war zones where people having incurred post-traumatic stress disorder, and they've shown very, very good results by exposing them and to those memories in using virtual reality. However, again, I've mentioned it already, but this is not something you give the headset to somebody and you say, well, I'll see you, just practice on your own. This is something that you do as virtual reality assisted therapy. Mm-hmm. So it's just a way, instead of using imagery, it makes it more powerful by immersing people in the situation, and it needs to be timed well and done together, the time that the person can can work with it in a meaningful way. Yeah. So it's not meant to induce more trauma, obviously.
Exactly. And do you think that this approach, virtual reality assisted approach, could actually in a way speed up the process, because we know that psychotherapy lasts a very long time? Can this
so that's one of the things that for example, we found in one of our studies with psychosis. That's the field where I work most, is for young people who have experienced the first episode of psychosis and they find it difficult to go back afterwards to social situation. The number of sessions we have currently in the protocol that we are implementing in first episode teams is only 10 sessions instead of 20 or 25 sessions, because you can reduce the time by exposing people in real time and helping and coaching them.
Oh, this is really wonderful. Thank you so much, Lucia, for sharing your findings with us, and congratulations on such wonderful, wonderful discoveries and combining VR with MRI. It's really great. Thank you so much.
Thank you. Bye.
Okay, welcome back everybody to our event, Neurotechnologies, Current Developments, Applications and Technical Issues. For those of you who are joining us right now, in the first part of this event, we have discussed transcranial magnetic stimulation, DBS, and so on. And now we are moving towards induced pluripotent stem cells, or short iPSCs, and their use in basic neuroscience as well as in clinics. We now have in a row 3 short talks, followed then by a keynote from Viviane Tabar. But first, I'm very happy to welcome Dr. Ivo Liberum from the Center for Neurodevelopmental Disorders here at King's to tell us something about stem cell-based models of neuromuscular diseases. Ivo, thank you for joining us today.
Yeah, thank you, Marija, for inviting me, and everybody for virtually coming here. So I would like to talk about new methods, all based on human iPSCs, that allow us to study neuromuscular diseases in ways that wasn't possible before. I try to go page. Oh, okay. Now it works. Okay. So I'd like to start with introducing some terms which I'm going to use during my talks. Motor neurons, which you see here labeled in orange, these are the cell bodies, and yellow, these are the axons, are the outward layer of the central nervous system. So they connect the brain to muscle, and they form, as you see here in the in this magnified image, they form these button-like connections to myofibers, shown in dark red, which are called neuromuscular junctions. So these are chemical synapses using the neurotransmitter acetylcholine. In some medical conditions, such as amyotrophic lateral sclerosis or ALS, these connections between the nervous system and muscle weaken, and this leads to paralysis and eventually the death of the patients due to respiratory failure within 3 to 5 years. A minority of cases of ALS, about 15 to 20%, have a known genetic cause, and these are called familial. The majority of cases called sporadic are probably caused by a complex interplay of genetic and environmental factors. One of the reasons why ALS is difficult to study in patients is shown in this scheme here. So while we have been studying ALS for decades and a number of genes have been identified that are linked to the disease, we don't completely understand the disease process itself. So one of the things we don't understand is whether, as shown in the top panel, the initial insult is within the CNS, and then this would lead to the degeneration of the cell body and eventually also retraction of the axon. Or if alternatively, the initial insult is at the axon, and the death of the cell body is only a consequence of denervation and loss of nerve muscle connectivity. The study of most ALS patients is not informative because you would then study them at a late stage of the disease, at a stage that resembles the image on the right, and that will not tell you the sequence that led to this degeneration. So in order to look at human neuromuscular circuits and study neuromuscular disease in a human-like model, we have developed a number of tools, the most important of which is the direct derivation of motor neurons from pluripotent stem cells. So in this example, we see derivation of motor neurons from mouse ES cells, but obviously the same technology also works for human ES cells and human iPSCs. You would start off with undifferentiated pluripotent cells, these are cell clusters on the left, and then through a neural progenitor intermediate and the morphogens retinoic acid and sonic hedgehog, which also program motor neuron identity in embryos, you can derive within a week in mice and within 3 weeks roughly in humans these cell clusters called embryoid bodies, which contain a decent number of motor neurons. And these motor neurons are then often labeled by specific genetic reporter constructs such as HB9 GFP. A second tool that turned out to be quite essential for the study of in vitro neuromuscular circuits is called optogenetics. So optogenetics relies on light-gated ion channels, such as channel rhodopsin 2, which can be genetically engineered into motor neurons, or well, any cell type really, and which upon light illumination lead to a net influx of positively charged ions, such as sodium, and depolarization of the cell membrane. And in neurons, this will lead to the firing of action potential, as you see here in this trace on the right side. So in the center, you see co-cultures of astrocytes in red and channel rhodopsin positive motor neurons in green. So this is essentially the most basic type of motor neuron disease model you can have. There has been an evolution over the last 20 years of these models from relatively primitive mixed cultures of motor neurons, interneurons, and God knows what cells, shown here on the left. So this is essentially where we started with neuromuscular disease modeling. These systems have become more refined. So the next step was to isolate neurons, motor neurons, and astrocytes, and have defined CNS-like co-cultures. This has then evolved into culture systems, initially unordered culture systems that also included myofibers and the connection between motor neurons and myofibers called neuromuscular junctions. And eventually the last step then placed all these cell types into compartmentalized tissue culture devices, so you could mimic the distinction between the CNS and the periphery with myofibers. This here shows an example for essentially step 2, so astrocytes and motor neurons. The experiment carried out by Fred Gage's lab. So in this experiment, they would take ALS-like astrocytes, which carry a disease gene called SOD1, and co-culture them in a multi-well format with motor neurons, human ES cell-derived motor neurons. And this co-culture system allows you now to expose these cultures in a very defined environment to chemical libraries and candidate drugs. One example is shown here. So when these cells are cultured for 2 weeks, motor neurons are perfectly fine. So the red label here is a motor neuron specific marker. The green label is a label for all neurons. However, if you expose the motor neurons to mutant astrocytes, then a non-autonomous effect leads to the death of motor neurons. So this was known before, but in this experiment, the Gage group then added a candidate drug, an antioxidant called apocyanine, and showed that you could rescue the disease phenotype and allow motor neuron survival over the 2-week culture period. I'm not saying that antioxidants are necessarily the cure-all for ALS, but this does show that you can get meaningful results with these in vitro systems. The next refinement, which I think is a really important step, and this was a pioneering paper in the field, was carried out by Ozaki and Kam. And they took the culture system to the next level by incorporating myofibers. Myofibers are really difficult to culture because they are contractile. I didn't really appreciate what that meant when I started these kinds of experiments. And in this culture system, you have 2 compartments. You have one here where you would place a spheroid that contains motor neurons, and a connected compartment that will contain a myofiber construct suspended between 2 flexible pillars. And with time, the motor neurons will grow axons and connect to these to this myofiber construct with neuromuscular junctions, which you see here. So these purple structures here on the left side are acetylcholine receptors, and you get connection of motor neurons to these neuromuscular end plates. So the motor neurons also express channel rhodopsin, which allows us to illuminate and activate them. And if you do that, you can then measure myofiber contraction as deflection of these flexible pillars, which is shown here on the right side. If the motor neurons carry a disease gene, in this case, mutant TDP43, you see marked here with red arrows some failures of contractions, and also the amplitude is lower than it is normal. And this phenotype can be rescued by single drugs or by a combination of drugs, as you see in the middle and the lower trace here. So my group essentially wanted to build on the advances by Roger Kam and colleague, and move these co-culture systems that contain neuromuscular junctions and myofibers to a multi-well format, which isn't really possible with the initial devices. So in order to do that, my student Pete Harley first developed human iPSC lines from disease backgrounds and controls that would contain magnetically sortable motor neuron markers. So you see that image here on the left. We can magnetically sort these motor neurons more or less to purity. They also contain channel rhodopsin, so we can activate them with light. And on the right side here, you see an initial characterization of the electrophysiological phenotype. So as you would expect from previous publications by Gareth Miles, for example, the disease motor neurons shown here on the left side in green are hyperexcitable during the early phase of the culture. And therefore, we were quite confident that we could that we did see a disease phenotype, and we could now move on to a neuromuscular co-culture system. The other element we developed in order to do drug screen on neuromuscular junctions, essentially, was carried out together with a material scientist at UCL, Wenhui Song, and another of my students, Amy. So Amy developed an elastomer nanofiber, a scaffold for stabilizing myofibers, which you see here on the top. So this is a composite elastomer with hard and soft segments. She would electrospin this into an aligned nanofiber scaffold and then seed iPSC-derived myofibers on top of that. And these myofibers could be entrained by optogenetics. And we showed, as you see here on the lower right side, that on a non-nanofiber control substrate, these cultures would deteriorate with time because the myofibers simply snap. Whereas we do get stable neuromuscular circuits with a nanofiber scaffold. So these two technologies, the motor neurons with channelrhodopsin and the myofiber culture system, was then combined into a multi-well format. So you would have nanofibers on the bottom, myofibers on top, and then a spheroid of astrocytes and motor neurons in the center, from which you would then get radial outgrowth of motor axons and the formation of neuromuscular junctions, which you see here in this high power image. So blue is myofibers, green is motor axons, and then red and orange is pre and postsynaptic structures. So you can use that to evaluate NMJ formation and disease phenotypes by high content imaging, which we did here. So you see in the middle image, you have reduced motor axon outgrowth in TDP43 mutants compared to isogenic controls. We could quantify this, and we also saw that NMJ volume, and not shown here, NMJ numbers is dramatically reduced. The NMJs are defined as an alignment of pre and postsynaptic structures. So we are currently using this culture system, which we just developed, to carry out a pilot screen with compounds that target the neuromuscular junction and distal axons in collaboration with AstraZeneca. Yeah, this was essentially everything I wanted to present to you, and I'm happy to take questions.
Thank you very much, Ivo. This is incredibly interesting, and especially I would say for me, because what many people don't know these days, I kind of started my PhD with NMJs, with neuromuscular junctions. So it was kind of really nice to go back in those great times when I was a carefree PhD student. There are a few questions that we have got, and maybe I can just try to summarize the topic. Mm-hmm. So there are questions about specificity / reproducibility of motor neurons derived from iPSCs or ESLs, mm-hmm. Because we know that even when they are derived from the same patient or after kind of passages over time, they gain or lose some properties. How do you deal with that?
Yeah, so that is a very good question. It's even worse than that. So even if you have the same passage number and the same cells and the same reagents and you do the differentiation 3 times, the motor neuron number might vary between 15%, 50%, and 10% in the next experiment. And then you have a mix of undefined other cells, progenitors, other neurons, with, again, variable percentages. So this makes it really difficult to do a drug screen because you can't compare one culture to the other. So in order to get around this problem, we developed this sorting method. So our motor neuron cultures always have 95% of motor neurons. It doesn't matter whether the initial culture had 10% or 50%, because we can adjust this by sorting the cells. And the same also applies to the astrocytes. So we are sorting both populations separately, put them together either in spheroids or in 2D cultures, and that dramatically increases reproducibility. And we can, we have done this already, so we can expose them to candidate drugs, dicrostatin, for example, and do get the same result reproducibly. But reproducibility with a traditional culture system where you avoid cell sorting is a big problem.
So everybody interested in that topic, please contact Ivo directly for the recipes and tips and tricks. And just one last thing, because we are again running out of time. Do you also use genome editing, maybe?
Yes. So we are now using genome editing to generate isogenic control. So we did have wild type ESL motor neurons as a secondary control, but I think the gold standard should be that you don't want to have a normal motor neuron from an unrelated person, you should always correct the gene mutation by CRISPR-Cas9 because that will help you to distinguish the effect of modifier alleles, for example, and the original mutation. So yes, we do use genome editing to introduce all these transgenes like channel rhodopsin markers and also correct mutations.
Thank you very much, Eva. It was a very instructive talk. And again, to the audience, any questions about Eva's protocols, please do contact him. It seems absolutely fantastic. Now we have to move on. And our next speaker is Eric Dine. He's actually still a PhD student at the Kent State University in Ohio in the States. And he will tell us something about how to use nanoparticles to deliver or to assist therapies for neurodegenerative diseases. Eric, thanks for joining us today.
Hi, how are you doing? Can you hear me?
Yes.
Perfect. Yes. Hi. So I'm done with my PhD. I'm at the NIH now in the United States.
Excellent.
So
congratulations.
Thank you so much. So yes, I'm going to talk today about using nanoparticles to target beta amyloid fibrils. This was my research I did for my dissertation and research that is currently ongoing at Kent State that I feel very strongly about. So I have no disclosures, and the title of my talk is Nanoparticle-Mediated Thermal Disruption of Fibrillary Beta Amyloid Proteins. So implications of using nanoparticles and particularly magnetothermal energy to disrupt proteins in neurodegenerative diseases. This is not just going to be about Alzheimer's. This could be applied to other diseases such as ALS with alpha-synuclein and other proteinopathies. So in the case of Alzheimer's disease, we deal with the amyloid beta protein, which is formed through this fibrillary pathway by this, the cleavage by beta secretase and then followed by gamma secretase give this peptide that is prone to aggregation known as beta amyloid, whereas there's a non-fibrillary pathway when it's cut by alpha secretase and then subsequently by gamma secretase, which is readily cleared. As these monomers are found in the cell, they begin to aggregate into oligomeric forms and then fibrils in the end. And these are classical signs of the Alzheimer's disease where you see these fibrillary A beta tangles. So what happens here is when we see the beta amyloid increase, we see neurotoxicity. So we had to reproduce this in our lab. So we had come up with a method to produce fibrils, and then we checked them looking at 0-hour incubation and then 16-hour incubation at 30 degrees Celsius, and showed that we do indeed have this change in size between these small and large amyloid beta fibrils. And we confirmed that using dynamic light scattering analysis, as well as both TEM, transmission electron microscopy, and then scanning transmission electron microscopy to show we do indeed form large plaques. So ultimately, the larger the beta amyloid plaque is, the harder it is cleared to be cleared by microglia, which are the phagocytic cells of the brain. So here we show that in the small beta amyloid, we had stained the beta amyloid with THT and then looked at the rate over time. And by 48 hours, we see a decrease in the signal of small A beta when we looked at the microglia, whether or not they had positive signal for A beta compared to the large, where there was not much of a change after 48 hours. And then we also used flow cytometry to confirm this. So ultimately, we also see an increase in pro-inflammatory cytokines, and we interestingly found that there is a change in pro-inflammatory cytokine gene expression based on the size, comparing the 0-hour and then the 16-hour A beta, showing a significant increase in inflammatory signaling using qPCR analysis. So ultimately, this shows that phagocytic capacity is decreased as the beta amyloid fibrils become larger, and then the pro-inflammatory gene expression is increased. So we were interested in finding a non-pharmacological approach to disrupting these beta amyloid plaques. And this is not to say that beta amyloid causes Alzheimer's disease. That's a huge thing. We don't necessarily know yet whether it's beta amyloid, whether it's tau, or some other something else that is changing the progression of this disease and is rooted in the etiology of Alzheimer's disease. However, this principle is we can use magnetic nanoparticles to generate heat, and then we can localize these nanoparticles close to the beta amyloid plaques. And the ultimate goal would be to denature the protein and then break it up so that they're smaller, and then beta amyloid can be cleared by microglia. And so in order to do this, we use an alternating magnetic field, which is in a simple term, we add an alternating magnetic field, which is going to cause the nanoparticle to move. And through Neal relaxation and Brownian relaxation, this is going to generate heat. And depending upon the intensity of the field, this will generate different degrees of heat. So ultimately, the proposed mechanism would be that we will add these nanoparticles to the beta amyloid, apply them to the alternating magnetic field, and then that will cause the beta amyloid to fragment into smaller pieces, which then microglia will be able to clear. So, and here's ultimately our proof that we were able to co-localize the nanoparticles to the plaque. So this here is our large fibrillary plaque, and then here are these small dots. Those are the nanoparticles, and we did not have to target them for this stage. At this point, there is no ligand or targeting molecule bound to the nanoparticle, but that is something that we can most certainly do. So ultimately, we show that here, without nanoparticles, without AMF, there's no change in the fibril. We add AMF, the fibril does not change, and then with just nanoparticles alone, there's no change in the fibril. However, after the application of magnetic nanoparticles as well as AMF, we see that the plaque now has been fragmented into smaller pieces. So ultimately, we tested to see whether or not this would change the clearance and the inflammatory gene expression, which indeed it does. Here in the red, we show this is our unstained control using the same fluorescent signal THT, and then we have our negative control, which is just beta amyloid unstained, and then we looked at 3 different trials with stained beta amyloid following AMF exposure for 15 minutes, and indeed, by 72 hours, we see a decrease in the expression of the beta amyloid, which we took as indication of clearance. And interestingly, we also showed that this decreased pro-inflammatory gene expressions. So we were very happy about this, and we were also interested to understand how this may impact the cell. So using scanning electron microscopy, we looked at the minor processes, the major processes of microglia, as well as the cell perimeter, and showed that indeed the nanoparticles alone increase the size of the cell body of the microglia, which is indicative of activation. And then the AMF had a significant decrease in the cell body size, as well as we see an increase in the process length, as well as the major and minor processes, which is quite positive since this is indicative of surveilling microglia. When the cells have their processes out, we consider them in a more resting, non-inflammatory state. And we also used A-beta to understand the effect of A-beta alone, and again, see that the cell is activated as well. The processes are retracted. So we see very interesting data here that supports that this heat treatment may actually be promoting which is, I mean, it's helping with the case of neuroinflammation as well as cell toxicity. However, we're wondering if this actually changes the phagocytic processes. And additionally, the application of magnetic nanoparticles did not induce or reduce inflammatory gene expression. So, as I mentioned previously, we're interested whether or not that NMP-AMF is going to change the function of the microglia. So we looked at the effect of AMF by looking at bead uptake. So we had uniform beads that were 1 micron in size, and we looked at the number of beads that were endocytosed by microglia and showed there's a significant increase in phagocytosed beads following AMF exposure. And this is without A-beta. And so this suggests to us that there may be a mechanism such as chaperones. So in order to sum this up, I went kind of fast because I want to get you guys back on time, but in general, the amyloid precursor protein is cleaved by beta and then gamma secretase, forming these fibrils. These fibrils are neurotoxic to the neurons and also to microglia. And without any intervention, this is going to decrease the phagocytic capacity of microglia, as well as increasing the pro-inflammatory gene expression, also done by microglia. So by targeting the beta amyloid plaques with magnetic nanoparticles and then applying AMF, we're able to fragment these beta amyloid fibrils into something smaller, which then has been shown to decrease the pro-inflammatory gene expression as well as increase phagocytic clearance efficacy. And while I did not show the data here for time, we have supporting data that this is actually due to chaperone-mediated autophagy, specifically through heat shock protein 70 and 90. So when we see an increase in heat shock protein 70, I'll just go through this briefly, and we see there's an increase in both macroautophagy and chaperone-mediated autophagy pathways in both LC3 and LAMP2, which this LAMP2 increases, and then that recruits the beta amyloid to an autolysosome, which then will hydrolytically degrade it. And by testing an HSP70 inhibitor using apoptosol, we showed that there's a drastic decrease in LAMP2. However, the macroautophagy pathway, there's a decrease in LC3, but not significant. So this may be the due cause and the reduced A-beta clearance following heat shock protein expression. I want to give a shout out to the 4 undergraduates that I was very fortunate to work with: Sandy, Megan, Matthew, and Reagan, who have all done amazing things, and I'm very proud of them. And we actually 2 weeks ago had a paper in the Journal of Comparative Neuroscience or Neurology come out. So I'm very proud of my undergraduates. They're excellent. And then I want to, of course, thank Dr. Kim, the PI, and Bing Yu, and then our collaborators, Thorsten Lars Schmidt at KSU, as well as Songping Wang, and then Junfeng Li, who was instrumental in a lot of the chemistry behind this project. And if anybody has any questions that I'm not able to answer today, please do email me at edine@kent.edu, and I'll be happy to answer all questions. Thank you.
Thank you very much, Eric. And again, as I said before, I cannot find this virtual applause up, but a big applause for this. This seems very, very promising. Thank you for presenting your research today and sharing it with us. We have a couple of questions for you. So let me just see what I have written down. So you said, in this approach, you're generating heat. So, in the beginning, when optogenetics was just starting, heat was also a major issue. So how do you control for not just really side effects, but off-target effects? So heat, damage of surrounding tissue, unwanted effects.
That's an excellent question. And yes, proteins are degraded around 43 degrees C, and there is also the possibility that nanoparticles will interact with other cells aside from the microglia, especially knowing that microglia love to eat up the nanoparticles. So we want to localize these to the microglia themselves. We want to localize these to the beta amyloid plaques. So there are ways to do that using liposomes and targeting modalities. But in terms of off-target effects, we did test the effect of uptake of nanoparticles at different concentrations by microglia, and then looked at their morphology as well as their cytokine expression. And we did not see any effect on microglia aside from a change in the surface topography, showing more. We believe them to be phagocytic cups, so it's actually might be helping the cell, but this is also got to be controlled by the material of the nanoparticle as well as the amplitude that we run the machine at, because the higher the power, the quicker the heat is generated. We had tested several different intensities and showed that at 1.2 millihertz, it produces a lot of heat at a very fast. I think it's dangerous around 7 minutes, but we had done this around 400,000 kilohertz, or sorry, 300 or 400 kilohertz. And the cells seem to tolerate this really well. And part of my research was going to be looking at the effect on neurons, but COVID kind of destroyed that. So I wasn't able to do that. Yeah.
Okay, thank you. But this seems very promising. And one last question, because again, in the interest of time. Sure. So I'm not sure if you have already maybe answered that, but it seems that you're also activating microglia, right?
Yes.
So somebody actually in the audience asked, activated microglia can do lots of damage in the brain. So how do you deal with that?
Well, so the nanoparticles themselves that we use do not activate microglia. It's been shown that nanoparticles larger than 50 nanometers, those will indeed activate microglia. However, our sample, we used magnetite nanoparticles that were 25 nanometers in size. As well, we showed that the heat actually attenuated. We showed that the fragmentation decreased pro-inflammatory gene expression and decreased cell body perimeter size, and so that would be active. That would be indicating that there's a resting microglia. So taking activated microglia by microglia being activated by the beta amyloid or by cytokine release, pro-inflammatory. This actually reversed that, so this is beneficial. However, microglia do need to be activated to be phagocytic. That's the common thought, that indeed, if we showed resting microglia, smaller cell body, long processes, they may not be as efficient at reducing the beta amyloid burden because they will not be phagocytosing beta amyloid. However, the beta amyloid is a potent inducer of activation by microglia, and this reduces that. So it would indicate that microglia become activated, that the heat may be promoting heat shock protein expression or some other chaperone that maybe we haven't identified yet that induces the clearance of beta amyloid, which then decreases microglia activation. But microglia have to be activated in order to be phagocytic.
Yeah. Thank you. It's all such a complicated process. You know, you get some, you lose some, and then you have to deal with all of this. Thank you very much, Eric. And now we are moving to our last speaker in this session of short talks, and this is Professor Jack Price, again from King's. And Jack will tell us something about pluripotent cells and the future of clinical neuroscience. Jack, thank you very much for accepting our invitation to join us today. Jack, are you with us?
Sorry, try again. Yes, well, thank you very much for the invitation to join this interesting conference. I've enjoyed it very much up till now. So what I'm going to do is, well, first of all, I'm going to show you my screen. Whoops, that wasn't what I wanted. I wanted that one. There we go. So I'm going to, obviously a very short talk, I'm going to whiz through just a couple of applications of pluripotent cells that I think the broader neuroscience audience will be interested in the context of how they're going to impact neuroscience, particularly clinical neuroscience and psychiatry over the next few years. First, let me just be absolutely clear that we know what we're talking about with what pluripotent cells are. So pluripotent cells really now are sort of two types. There's the embryonic stem cells, which, as you know, are derived originally from the inner cell mass, breakthrough technology that really gave us access to human embryonic stem cells some decades ago now. And at this point, I just want to give a brief to my old colleagues at the UK Stem Cell Bank, where they've got about 150 human ES cell lines that are sort of freely available, including 39 clinical grade lines that can be used for some of the applications that I'm going to talk about later. And then, of course, the other type of pluripotent cells that we're all familiar with, and Ivo has spoken about, are the IPS cells that we now have access to, where one starts with a source of cells. It could be any source of cells. In our lab, we've mostly used hair-derived keratinocytes, and they can be turned using the 4 Yamanaka factors into colonies of cells, which could be expanded into lines that are essentially functionally indistinguishable from the human embryonic stem cells I just mentioned. And of course, they're pluripotent. They can turn into lots of things like neurons, the main thing that we're interested in. So, and the point I want to start by accentuating is that the real impact, the first impact of these technologies, this access now to pluripotent cells, is that we can now generate bucket loads of human neurons of any type. These are human cortical neurons. There's about, I don't know, 10 to the 7 cells in this culture. And if you like these cells, well, you can have another 10 to the 7 absolutely identical tomorrow. And this is a sort of an access to human neural material that we've simply never had in the past, and allows us to avoid a lot of the issues we've had with trying to translate from animal studies in the past. So I'm going to speak very, very briefly just to update you on what I think the main impacts are going to be in 2 areas where pluripotent cells I think are going to be important. One is disease modeling, and the other is regenerative medicine. So to start with disease modeling, what I really want to introduce you to is a work stream rather than a very specific outcome or a very specific set of experiments, because I think a generic work stream is emerging in the study of neurological and psychiatric disorders using pluripotent cells as the starting material. And that's really as following. Quite frequently, it starts with a genetic model. So Ivo spoke a little bit about ALS. That's one of a number, obviously, of neurological disorders which have strong genetic associations. Using a genetic model, it allows us to identify a phenotype or a number of phenotypes. And this then, using the stem cell approach, can be expanded into generic models of disease, and those really give us access to mechanism. So let me just explain how this works with reference to a system that we've used in our laboratory over the last few years. So we were interested in the sort of etiology, the developmental neuropathology of autism. And as many of you will be aware, SHANK3 is one of the major genes associated with the etiology of autism. And we made iPS cells from a number of individuals, autistic individuals who had mutations in their SHANK3 gene. And to cut a very long story short, what we found was, sure enough, there was a synaptic phenotype in these cells, which is precisely what you'd expect. SHANK3, of course, being a prominent synaptic protein. But somewhat surprisingly, we also found a much earlier phenotype. Indeed, as soon as we generated postmitotic neurons from these SHANK3 mutated iPS cells, we saw a morphogenetic phenotype, but by which I mean the cells immediately that were postmitotic started to grow a different size and shape than the control counterparts. So that gave us, starting with this genetic model, that gave us a phenotype. We could then expand that into a more generic model of disease. Now, autism obviously has genetic antecedents, but it also has environmental risk factors as well. And one that you'll probably be aware of is maternal immune activation. So if mum gets a very severe viral illness during her first trimester, she has a somewhat increased risk of having a child who will subsequently receive a diagnosis of autism. And this was also a system we were able to reproduce in an iPS culture, not now starting with SHANK3 mutated cells, but rather starting with wild type derived iPS cells. And basically, again, to cut a very long story short, we were able to find exactly or a very similar morphogenetic phenotype in cells that went through this model that was exactly similar to what we saw in the SHANK3 mutated cells. In other words, we'd started from a genetic derived phenotype to one that seemed to be much more broadly applicable to the condition we were looking at, namely autism. And using this system, we were then able to go on and look at mechanism. And again, I don't really want to go into great detail. The work, if you're interested in the details, are in these two published papers. But basically, to cut a very long story very short, we were able to identify the mechanism in this model, whereby this change in morphogenetic development was generating, and it worked through PML nuclear bodies, which in turn had an impact on transcription, particularly of MHC class 1 genes, and this affected neurite outgrowth. So as I say, I'm concentrating rather than on the details on the work stream, how we went through this genetic model to a phenotype to generic model and on to mechanism. Now, this is becoming broadly used, and others are using it to go one step farther. That wasn't appropriate in our particular model, but a further step that can be taken here now is that can be used as a model for drug discovery. And there are a number of examples of that out there in the literature now. Just to give you one, and Ivo's already told you a lot about ALS. This is work from Clifford Wolf and Kevin Egan's labs in Harvard, where they used basically a very, very similar work stream to identify a phenotype and a mechanism underlying a phenotype that related to motor neuron hyperexcitability. And then they could use precisely this cellular model to go and do drug discovery studies. And in fact, they found good candidate molecules which are now in clinical trials. So the general point is this, that I think this is a work stream, this is an approach using pluripotent cell models that are going to impact neurology and psychiatry research really quite heavily over the coming years. And I think that broadly the field needs to take cognizance of this. Now, the other point I want to make about, before I move on from this little bit of the talk, is that we concentrate on pluripotentiality when talking about iPS cells. But in many senses, that's not the most interesting feature. I mean, we've had cells that could give rise to neurons and glia for many, many years. But what we haven't had until these pluripotent cells came along are cells which really could undergo proper tissue histogenesis. These cells don't just make neurons and glia, or indeed, of course, a much broader range of cell types. They can actually build tissue. And I just wanted to draw attention to that by just showing you a couple of pictures. This is what you get in a monolayer culture if you neuralize iPS cells. You start to see them starting to try and make a neural tube in culture, and they make these what we call rosettes for reasons that are obvious, which are a sort of series, if you will, of cross sections through a forming neural tube, and they form a polarized neuroepithelium. And the red cells here are neurons that have migrated out of the neuroepithelium, just as they would during development in vivo, and lie outside of the neuroepithelial layer. And you can push this farther. This is a piece of cerebral cortex, if you will, that we've grown in our lab. And again, you can see the polarized neuroepithelium, and the red cells here is basically a baby cortical plate forming in vitro. Others have taken this farther. Many of you will be aware of the work of Madeleine Lancaster, who really started this off with these mini brains that she was able to grow in culture. And people have taken it much further now, and we have really quite sophisticated ways of growing 3D cultures of neural cells, of neural tissue. And people are also now starting to play with growing 2 different types of neural tissue, be it thalamus and cortex or striatum cortex, and wiring them up together in vitro. And the power of that, I'm sure, is immediately obvious to you, and it's something we're going to increasingly come across as we go forward in this field. So that's all I want to say about disease modeling. Let me just draw attention to a couple of ways that pluripotent cells are starting to impact regenerative medicine. Now, regenerative medicine, stem cell therapies and the like, have of course been around in brain science for probably 30, 40 years, but they've always struggled in a number of regards. And one of the major areas where they've struggled is getting any sort of consistency in the therapeutic product. Typically, the cells have been a mix of cells, very poorly characterized, which has meant that functional assays and product release assays have just been very, very poor. And regulators have been very generous with us over the years in letting us proceed with medicines that are really very poorly characterized. So pluripotent cells really allow us, I think, to start to address those and get an accurate product specification, true scalability, and a product control that really hasn't been possible here to now. So let me again give you just one example or a couple of examples of this. So you'll all, I'm sure, be aware that a favorite target of regenerative medicine in the nervous system has been Parkinson's disease. And again, you'll all be aware, and I'm sure we're going to hear more about it in the next talk, you'll all be aware that for many years people were taking a fetal ventral mesencephalon, the source of the dopaminergic neurons in the embryo, in the fetus, dissociating that chunk of tissue and using that as a therapeutic material. And you'll all also be aware that was an enormously unsatisfactory source of material for a whole series of reasons, not least that it was a horrible mix of different cell types. And also the results as a consequence are enormously variable. But what's happening now is 2 technologies are coming together. The first, sorry, I've just lost that. The first is single cell transcriptomics. And what I'm showing here is that this piece of tissue now, and this is a piece of work that's come out of Karolinska in collaboration with Cambridge, have been able to do single cell transcriptomics of this material that previously we used as just a whole cellular mix, and identify each of the different cell types that make up this material. And sure enough, there turns out to be 20-odd different cell types in there. And of course, only one of them is really the cell type you wanted. And now that's been identified using this method, that knowledge can be plugged into a cell production method, starting with the stem cells, starting with the pluripotent stem cells and driving them towards exactly the cell type you want, thereby eliminating a huge amount of their variability in the starting material. And going alongside of that is other efforts to characterize in much more closer detail the final cell type that you're looking to derive. So this is a piece of work that you can see the publication here, characterized cortical cell types. And it turns out that in the mature cortex, there are several hundred cell types, where previously we thought there were maybe a dozen or maybe a few more. So this is an enormous opportunity, but it's also an enormous challenge, because it's putting under pressure on people who are trying to do regenerative medicine for brain disorders to firstly start with a much more precisely characterized starting material, and then use a protocol to derive a much more precisely characterized final material, final product. And so I think that there won't be a pause in this approach immediately, but what there does need to be a much more precise thinking out of where we want to go with regenerative medicine. And these are opportunities that have been allowed by these pluripotent cells. One more final example, I'm just very, very keen on this. This is work that comes from the Institute of Ophthalmology here in London, Lacruz and Pete Coffey's efforts. These are retinal pigment epithelium cells, which, as you know, are what sort of get lost and die in macular degeneration. The lab there have managed to generate these beautiful monolayers of retinal pigment epithelium to go into patients with macular degeneration and to generate this really engineered patch that can be introduced into the retina using this device, this companion device, and is now allowing a really much more precise production of a therapeutic product and administration of a therapeutic product. And again, none of this really would be possible without really a really precise control over the starting material and the manufacturing process. And again, this kind of combination product is very much something that now I think starts to become available. Now we can use this precise starting material. So that's all I wanted to tell you about. And basically what I've told you is the potential of pluripotent cells in these two areas: unlimited human cells, enhanced construct validity of disease models, potential for drug discovery and histogenesis, and really panning into regenerative medicine where there's these accurate product specifications, scalability and production control. That's my time, so I'm going to stop. These are the people who did the work that came from our own lab that I've told you about. These are collaborators and funders. So thank you very much for your attention.
Thank you very much, Jack. I must admit, I love your last slide, collaborate or die. I just love it. And it's so true. I mean, the message is really true. We are quite, ahead of, above our time. So I will be very brief. I believe that there are some efforts, right, to also use iPSCs to kind of delineate, in autism, for example, you have certain genetic mutations which are shared, for example, with schizophrenia. So I know that some scientists are trying to use iPSCs to see if maybe mutation is enough to cause one phenotype and then maternal immune activation on top of it leads to another. Is that right?
Yes, I think that's correct. So once you start to derive these models, you can start to combine them. That's exactly correct. So you can look at the interaction of genes with environmental risk factors. And this is something that certainly sort of has existed in this kind of research for really some time, but mostly using mouse models and mouse cells. And as you're alluding to, we can really now do this with human material. And I think that's going to be enormously beneficial. Although, of course, the downside of any research on a psychiatric disorder like autism is you've got no behavior. How it relates to the behavioral syndrome is of course a challenge, but we have to address that.
But we need to take a step at a time. And what I personally like about iPSCs is that they're derived from a patient. Yes. So it's still kind of, we always model something. So there is a plus and a minus to that as opposed to animal models. So we really need, I think, everything to get more holistic picture. And one last question actually from the audience before we continue is, somebody asked if, related to this macular degeneration, if that treatment is already available in clinics?
My understanding is that they've done a small number of patients in a small clinical trial, and now they're expanding the clinical trial. That's my understanding from the last time I spoke to Pete Coffey. I'm not entirely sure where they're up to at this present moment, but it's certainly under development.
Okay, thank you very much. And I would just like to finish this by actually telling everybody, in case you don't know, and I'm sure many of you do, that recently, last year, I believe, Jack also published a great book called The Future of Brain Repair, a Realist's Guide to Stem Cell Therapy. So for everybody, if you would like to know more, please take a look at the book. Jack, thank you very much.
Yeah, well, thank you.
I would like to also take this opportunity to thank to all of our speakers today. We have come to the end of today's session. And just to quickly recapitulate what we have been hearing today about. So Basically, we have started with Freud. I remember that everybody still remembers the talk of Paul Scholpot about F and D. And we moved then from Freud to 21st century and the development of various neurotechnologies. We have seen the ways how neurotechnologies can improve human lives, and that actually they can act on a much faster time scale than, for example, all the strategies we have used before. However, there are certain ethical aspects that we still need to discuss when we talk about the use of neurotechnologies, and we are going to do that mostly today. This discussion, tomorrow. This discussion will be at the panel session at the end of tomorrow's session of this conference. We have also seen today benefits of bringing neurology and psychiatry closer to each other, kind of closing this gap into so-called neuropsychiatry that really benefits patients, especially FND patients, for example. And please join us all again tomorrow when we will continue this conference at 1 p.m. by looking more closely into various tools for analyzing and repairing the brain. So there we'll be talking about, for example, optogenetics. Then we will also move on into neuroimaging to see how we can use neuroimaging to parse heterogeneity in autism, which is a very important topic for many of you, I'm sure. Then we will move to spinal cord injury and new and novel neurotechnological approaches to help spinal cord injured patients. And at the end, we will have Dustin Tyler with us, and he will start the discussion on the ethics of the use of different neurotechnological approaches. And we will finish this conference then tomorrow about 4 p.m. by a panel session before that about all the applications that we have heard about and discussion of the ethics. Thank you again for joining us today, and we hope to see you all again tomorrow. Bye.